CUHK iGEM team, and so before we talk more about our project, maybe we could introduce ourselves. So my name is Allison, and I am a year 4 biochemistry major, and I am part of the wet lab team.
My name is Chang, so I'm an incoming year 4 in biomedical engineering. I'm part of the dry lab team.
I'm Jasmine, also majoring in biochemistry in year 4, and I am taking part in human practice and also in wet lab team.
So I guess before we start talking about a project, maybe we can talk to you more about the inspiration behind why we decided to do bioremediation for our project. So I think it's no surprise to everybody that there is an increasing amount of waste being sent to the landfills, and just in Hong Kong, for instance, the Environmental Protection Department is saying that they want to increase the capacity of the current 3 landfills that we have in Hong Kong. And as you can see from this, that currently the sustainable alternatives like paper products do not seem to help alleviate this kind of burden on our landfill capacity. So in fact, it is estimated that there are actually 400 million plastics and paper cups that are being consumed every year just in Hong Kong. And in a day, there are about 2,331 tons of plastic that are being sent to the landfills per day. And among the 2,331 tons is 285 tons of plastic that cannot be recycled. So you might be wondering why there is a continuation of so much plastic and paper products that are being put into landfills. And despite the growing efforts to recycle, we deduce 2 major reasons why this happens. And the first is the overflowing amount of mixed waste. So like the VLT that you drink, for instance, which are laminated with, if you didn't know, like some metals or resin or maybe even plastic materials, juice boxes or milk cartons that you drink in your everyday lives also are realistically quite difficult to recycle due to some of these mixed waste. So there are actually 2 main types of recycling that exist currently, and one is the single stream where everything is basically just put into one bin, and the second is a multi-stream. So currently Hong Kong generally uses the multi-stream. So we split our waste into metal, glass, plastic, and paper, and you probably see these bins quite a lot actually, maybe in schools or even the train stations. However, the problem with this segregation system is that there isn't an option where you can throw away mixed waste. And so because there is no option to throw away mixed waste, they are generally just sent to the landfill. So another issue that we also deduced regarding the recycling is the lack of public awareness on the proper ways to handle sort of mixed waste, or people are just like simply too lazy to recycle. So another issue that we found is, so besides plastic waste that we mentioned, also paper waste is a growing concern. So since 2020, there has been a growing increase in manufacturing paper waste products, and there has since been a 4% increase due to their presumed beneficial alternative to plastic production. And also many are unaware that actually producing paper products generates 3.5 times more greenhouse gases than manufacturing plastic packaging. So as paper product is expected to keep increasing, significant efforts are needed to sort of reduce the emission intensity of production by diverting attention from fossil fuels as an energy source and perhaps promoting some technological advancement that reduces the heat of the pulp and paper drying industry. So currently, the paper pulp industry in Hong Kong and other Asian countries like Singapore, for instance, is given limited opportunities to sort of expand their businesses in this industry. And we hope to sort of bring awareness to these paper-based alternatives that are not completely a sustainable option, and that the best option actually is just to utilize like reusable products and such as like tumblers or maybe like metal straws in our everyday lives. The motivation for our project stems from these three main categories: so the environment, social, and economy. And we hope that with our project we are able to contribute to the limited research in recycling paper-based alternatives, such as paper cups that are strongly coated with a plastic material called polyethylene, or in other words, PE. And paper cups is just one of the few popular bio-alternatives that highlight the lack of highly specialized recycling facilities or collaboration between different sectors, such as like the government and the other businesses and customers even. So we hope that to ease the collaboration of all these different sectors, that we can provide a more cost-effective technology that can be easily implemented to facilitate the effective recycling of this kind of waste. So our project aims to investigate these three aspects. So the first being to maximize the protein yield by producing a codon-optimized genes that ease the transcription process of our enzymes by adjusting the spatial distances between the RBS site and the start codon. And we will also determine our plastic degradation efficiency of PE and another material called PFA by measuring enzymatic activity and the optimal enzyme concentration as well, in order to undergo our biodegradation experiment. So I mentioned PFA. PFA is actually another type of plastic material, which is usually made in like food contact material. So a lot of the food packaging that we use contains PFA, and the material PFA itself can actually, when you eat the food from the packaging material, it can actually transfer to the substance of the food in a process called migration, and it actually has direct harms to our health. So the reason why we decided to investigate mainly these two products, besides the fact that there's an overwhelming amount of it in our products that we use today, but also that a lot of the iGEM teams that we noticed focus on degrading one type of plastic, and we believe that this is sort of like an unrealistic portrayal of what the current recycling industry actually is. So maybe we can talk a bit about what codon optimization is and why we decided to use it. So as we mentioned, so the codon optimization, it happens in like at a gene level, and we did this in order to increase our translation efficiency for protein expression level, and we use, in order to allow us to do codon optimization, we use a method that requires using Gen Smart Codon Optimization tool called GenScript. And another thing that we mentioned is promoting a better, protein expression. And another method to do this is making unique spacers in our biobrick. So the spacers that we mentioned actually stem from the SD sequence, and it's composed, as you can see, of like a urine-rich sequence at the 5' end, and which is complementary to the UCCU core sequence at the 3' end of that is attached to the 30S ribosomal unit. So the SD sequence, so the spacer that we mentioned, is actually located roughly in between the 10 nucleotides upstream from the start codon, the AUG. So how did we essentially determine what spacers that we're going to use for our project? So we found some studies that suggest that the 5 NT spacer and the 9 NT spacer are the best to use. And specifically in E. coli, the 6 NT spacer is the most optimal one. So based on this, we decided to make 3 specific spacers for our project, which we will discuss later on. So as I mentioned, we will create different spacers for our protein domain, and we will do this. After doing this, we will amplify these by PCR, and after PCR, we will add the prefix and suffix to our synthesized part to facilitate the construction of our plasmids or biobricks in order to create the genes of interest for our enzymes. The assembled plasmids will undergo a process called site-directed mutagenesis, and this process itself will allow us to create the different spacers that we mentioned. So we will be creating the 5, 6, 9, and 10 nucleotide spatial distances between the RBS and the start codon. The technique specifically is called PCR-based mutagenesis, where we will adopt a deletion type of mutation onto the forward primer, as you can see here, and whilst the reverse primer will be the same throughout the procedure. And last but not least, we will also measure the protein concentration quantitatively by including reporter proteins. So that's GFP, CFP, and YFP. And these reporter proteins will emit a fluorescent signal